Welcome everyone, Dr. Lindy here. In today's class, we're going to cover classifications of tissues. Now, if you remember back to the introduction to anatomy and physiology, we talked about how cells are created based on chemistry, right? That carbons and oxygen and hydrogen and nitrogen and phosphorus, they come together, or carbohydrates, proteins, fats, right? Chemistry comes together to make up cells. Cells come together and make tissues. Tissues make organs, organs make systems, systems make up the 11 systems make up the organism. So chemistry to cells, to tissues, to organs, to the 11 systems, to the organism. We're going to talk about epithelial tissue, connective tissue, muscle tissue, and nerve tissue. And I'll break these up into the 4 different sections. I'll have one separate video on epithelium. I'll have another video on connective tissue. Muscle is very short, so I will combine muscle with nerve. And then I'll have a 4th video doing an overview on all of these. I'll throw out some quiz questions that I've seen pop up in the past. And then what I'll do is I'll create I think I'm up to 5, a 5th video on skin. And the reason why we save the skin for last is because the skin is actually made up of epithelial, connective, muscle, and nerve combined. And I'll do a little bit of nails along with the skin. Okay, so again, when we get a group of cells that come together with similar structure and function, we're going to get 4 different types of tissue: epithelium, connective, nervous tissue, and muscle tissue. I use the word neural interchangeably with nervous. I just don't like to use the word very much, nervous. I'm the type of individual and healthcare professional that believes that there's a lot of power to words, and using the word nervous can have some negative impact on us. So let's look at epithelial tissue first. When we talk about epithelial tissue, this is the type of tissue that is a lining and a covering type of tissue. So when I think of lining and covering, we have something that covers the entire external surface of the body, right? We refer to that as skin, but anatomy and physiology, we know that skin has different layers. There's an outermost layer, epidermis. The word here, if I can, let me underline this for you. Let's see if I can get my pen here. So here, epi means above, and dermis is the skin. So epidermis. Now, if there's an epidermis, there'll also be a hypodermis and a dermis. So there's actually 3 different layers. So the epidermis covers the external surface of the body, but epithelium is also very important because it lines the body cavities and tubules. So what are tubules? In the word tubules, we see tube. So a tube could be like this, right? Any type of tubular system, which may be a blood vessel, maybe it's the intestines of the digestive system, maybe it's the trachea, maybe it's the bronchi, right? Maybe it's the reproductive tract. So anything that's a tube or a tract or a cavity is going to have epithelial tissue. Glands are also classified as epithelium because they do develop from epithelial membranes. Now, when we talk about epithelium, we were going to classify it and name it based on a concept of whether or not the cells that make up that tissue, are we looking at something that is 1 cell layer thick, or is it more than 1 cell layer thick? Meaning, if I'm looking at something under a microscope and I see something like this that is 1 cell layer thick, compared to something like this that may be several layers like this, okay? So if we see something like this that's one cell layer, we refer to the word as simple epithelium. And if we see something like this that's multiple layers, then we say that it is striated. So striated is multiple layers, simple is one layer. Now, the second thing we look at is we have to refer to, in its name, the shape of the cells. So in this particular case, and here, as well as here, we notice that I draw them as boxes or cubes. So then those would be referred to as cuboidal. But if they were taller, like this, with a nucleus, then we could say that they are columnar, right? We would say that they look like columns, columnar. But if they were like these flat, irregular shaped cells, like this, then we would say that they are squamous, okay? So one of the questions we have to ask us when we look at epithelial tissue is, when we look at it under a microscope, we go, all right, how many layers do we see? And then the second question is, what's the shape? And we said that they line cavities or lumen. So here is, let's say, a lumen, right, is the hole or the space. But we can have cells that go around this lumen, right, like this. Or we can have cells going around it that look like this, like columns, okay? So when we talk about, we name it based on the apical layer, we find the lumen, which is that gap or the hole or the space, and then we find the cells that are just adjacent to it, okay? And that's what we're using to look at the number of layers and the shape. Okay, so squamous, you see you have these flat shaped cells. Cuboidal are these square or cube shaped. And then columnar are these columns, like buildings. Now, simple, which we see right here, is when they're only one cell layer thick. And in all of these cases, we see that they're only one cell layer thick. But here in stratified, we see that they're multiple layers. Here, multiple layers. And here, multiple layers. These are flat, irregular shapes, so we would call them stratified squamous cells. Here, these are cuboidal, but they're more than one layer, so we would call them stratified cuboidal. And then here are columnar cells, so we're gonna, and because they're more than one layer, we're gonna call them stratified columnar. Now there's one other category that I want to introduce you to, all the way on the bottom right here, called pseudo-stratified. And this word pseudo means false. So there's something about this that gives it a false appearance of being multiple layers. Well, what is it that gives it this false impression? Well, if you look at simple columnar on the lower left, we see that all of these nuclei are at the same level. When we look at stratified cuboid columnar, we see these nuclei are at the same horizontal level, and these are. In fact, in all of these, the nuclei are even. But if you look at this one, we have some nuclei at this level, we have some nuclei at this level, and we have some nuclei at this level. So it gives it the appearance that it's 1, 2, 3 layers, but truly it is only one cell layer thick. So we call it pseudo-stratified columnar. And typically, where we see pseudo-stratified columnar epithelium, we will also see this brush border here, these, which are called cilia, C-I-L-I-A. Cilia are these little hair-like structures that can beat rhythmically. They beat rhythmically. In fact, we have, if this is your windpipe, and then we have columnar cells here and columnar cells here. I'm going to put a nuclei there. And then we have, let's say, another columnar and then another columnar. You kind of get what I'm doing here, right? Let's just go like this, and I'll make a bunch of columnar cells here, and then we'll make a bunch of columnar cells here. What we also have, let me change the color for a second, if I can. What we have, we'll make this blue. At the very tips, we have these cilia on both sides. We have these little hair-like cells that are going to beat rhythmically, and they line the windpipe as just one place. They also are found in the intestines. But then here is the lumen. This is where airflow is traveling. When you breathe in air, it's going in. And then here, and let's say here, and here, are other cells that are mucus producing. They're called goblet cells. These blue circles are goblet cells. And what those goblet cells do are they secrete mucus. So we're going to make this one, let's say, green. And we're going to put mucus along the cilia here. And mucus, we know, is sticky. So now when you breathe in air, if there's dust particles or debris, these goblet cells produce mucus that are sticking to the cilia, but the cilia are going to beat rhythmically upward. They're going to beat superiorly. So that now, as air is moving down to try and get into the lungs and debris is sticking to the mucus, since the cilia are beating upward, it can pull out dust particles and debris. So all you have to do is go and you cough it out and you're able to get the debris out. So that's the beauty of cilia. So wherever we see pseudo-stratified columnar, it's typically pseudo-stratified ciliated columnar, as we see in these pictures. So epithelium, their primary function is for protection, absorption, filtration, excretion, secretion, and sensory reception. So protection, if we think of the skin, right, that external layer, the epidermis, it protects us against any type of excessive friction or even bacterial invasion or chemical damage like acids from entering the body. And then again, we just introduced you to ciliated epithelium that we find in the respiratory tract to sweep up dust particles and any other foreign particles up and away from the lungs. Why and how? Because those cilia are beating rhythmically superior. In terms of absorption, we find them in the GI tract. GI is gastrointestinal for the stomach, small intestine. The cilia act as a sponge, if you would. It's designed to absorb vital nutrients. And if we were to draw, let's say here is part of the intestines. Again, I'm drawing it like a tube or a pipe. We are going to have columnar cells in here as well. And then we're going to have cilia, right? Those little hair-like structures. So the nutrients and the food that are broken down by digestive enzymes are going to get absorbed by the cilia. It pulls it inward, it pulls it, and eventually what it will do is, this is going to be a crude drawing, of course, but it will eventually be absorbed into your blood vessels, into your circulation, right? So all the food gets absorbed into here and eventually into your blood vessels so that you can bring all the nutrition to various parts of the body, okay? So that's the absorption. Filtration, because in the kidneys there are tubules, and the epithelium there is going to absorb certain nutrients, It can secrete and get rid of things, and the kidneys are a filter. So epithelium is really, really important in the kidneys. When you get into the kidneys, you will learn about the nephron. The nephron is the functional unit of the kidney, and you will learn that in the proximal and distal convoluted tubules, there are simple cuboidal cells. Okay, it's also for excretion and secretion. Excretion means to get rid of, right? So you can absorb things. The body is filtering, and after it filters it, we can absorb it or reabsorb it. We can get rid of it through urination. And then we have glands that can also secrete substances, okay? In terms of sensory reception, we have little hairs that are on the skin. And if something is crawling on the skin and stimulates one of the hairs on your legs or your arms or your head, it can notify your neural system that something is crawling on you. So that's a little bit about epithelium. Now, in terms of some function, I've seen this show up on certain quizzes, like certain Connect quizzes from McGraw-Hill in the past, where it was like a drag and drop. So what type of epithelium can function in diffusion, filtration, secretion, and protection? So when we see the word protection and diffusion, I think of the skin. Okay, in the skin, especially not just the skin, even your blood vessels have squamous cells. So when you think of diffusion, I think the skin may have probably not the best because the skin is actually stratified squamous, but it is involved in protection. When I think of diffusion, I think of this. I think of the airways, and then I think of your primary and secondary bronchioles, and I think of your lungs. So let's say this is lungs. There are cells that are simple squamous cells that are just one cell layer thick, and it allows for oxygen to pass through there, and it allows for carbon dioxide to go out. So there's this exchange between O2, oxygen, and CO2, and the only way these gases can move from one location to the other is through diffusion, passing through. So that's simple squamous. Simple cuboidal is absorption and secretion. There's the kidneys. And protection. Those are the kidneys. Stratified squamous is protection of the underlying layers. Here I think of the top layer of the skin because it really is stratified. It can have, I don't know, somewhere between 25 and even 30 layers of squamous cells. And I'll show you a picture of that when I cover the skin. Pseudo stratified columnar, there's the word pseudo, which means false. It functions in the movement of a secretion like mucus. Remember the goblet cells that I drew before in the trachea? So it can function the movement of secretions like mucus across the apical surface of the tissue, just like I showed you here, where we had the goblet cells producing mucus, and then because they beat rhythmically, they can move it along the apical surface. The apical surface, that top layer surface. Now, I didn't mention transitional, but transitional, think of a balloon, a deflated balloon. If you have a deflated balloon and you have a marker and you make a circle, or you make a letter E, like an alphabet letter E or an A, and then you inflate the balloon, doesn't it distort whatever it is you drew on the deflated balloon? So it can alter its shape. So basically, transitional epithelium can accommodate for distension and relaxation. Think of a balloon. Think of a urinary bladder. A urinary bladder can be relaxed, or it can be distended if it's filled with urine. So we have to give these epithelial cells the ability to change shape. Those are transitional cells. So I think of a urinary bladder, any type of organ that can increase in shape can alter its size. So again, we mentioned cilia before, but what I didn't mention are microvilli, but cilia are those long, slender extensions. We've seen them when we've covered the cell, because certain cells can have these cilia, which are extensions off the membrane, and it can move the cell, but it can also move secretions and fluids across the cell's surface, as we saw in the windpipe. But it also can happen in the fallopian tubes or the uterine tubes of a woman, where within those uterine tubes there are also cilia that are beating rhythmically to move the sperm towards the egg and the egg towards the sperm. Pretty neat how that works. Microvilli are these small projections that are primarily used for absorption. These microvilli are primarily found on the intestinal cells. We mentioned cilia are also found, but smaller hair-like projections along the intestines for absorption of nutrients and water. Those are called microvilli. And we can see by this picture that we have epithelium throughout all parts of the body. Here in the respiratory tract, we have the pseudo stratified, and remember I said wherever we see pseudo stratified, we'll see the little hair, so pseudo stratified ciliated columnar epithelium in the respiratory tract. Along parts of the gastrointestinal tract, but in the upper GI, in the esophagus, in the throat, we have multiple layers here to here. You see how we have several layers of these flat, irregular cells? Those are squamous cells, but they're stratified. So we find stratified squamous in the esophagus because you're eating food and the lumen of the esophagus can be scraped by food. It needs protection. No different than the skin needs protection. If you were to scratch yourself with your nail and it was only one cell layer thick, you'd bleed. So it's got to be 25 to 30 layers thick. In the airway of the lung, where we have alveoli, that's where we have more squamous cells, but these are simple. It's only one cell layer thick. That's where we have the diffusion of oxygen and CO2. That's the exchange at the apical surface of the epithelium of the alveolar cells, which are the air sacs. In the kidneys, okay, here is a lumen. So something is traveling through there, probably filtrate or urine. And then around it, we have these one cell layer thick of square cube cells. So it's called simple cuboidal. And then within the intestines, we can have one cell layer thick. We see that the nuclei are at one level, so it's simple, and they're columnar shaped. Simple columnar epithelium. So again, based on the layers, they could be simple or stratified. If it's one cell layer, it's simple. If it's multiple, it's stratified. If they're flat, irregular shaped, it's squamous. If they're cube shaped, cuboidal, or they could be tall columns. And then we mentioned the pseudo stratified. They're called pseudo false because the nuclei are at different levels, so it gives it this false appearance that it's stratified when it's really not, and oftentimes we will see cilia with them. The transitional, that was the balloon that I mentioned, how it can be distended or relaxed in parts of the body like the urinary bladder, the ureters, and even parts of the urethra. The cells can vary in shape. Again, the three different types of simple epithelium are simple squamous, simple cuboidal, and simple columnar. The different types of stratified are stratified squamous, stratified cuboidal, and stratified columnar. Then we have the pseudo stratified, and we have the transitional. Those would be under stratified. Okay, this is a good place to take our first break, but when we come back, we will start connective tissue.